And without further ado, I will turn it over to Mr. Bruce Bassett for introductions. Enjoy the class.
Well, hey there. My name is Bruce Bassett. In addition to being coordinator for this class, I'm also the chair of the Science, Medicine, and Technology Subcommittee of Curriculum. And I am delighted to have the opportunity to be with you and to have had a chance to learn just a bit about our topic today. Our presenter is Dr. Kristen Dittenhafer-Reed. She earned her Bachelor of Science in Chemistry from Hope and a doctorate in biochemistry from the University of Wisconsin. She also did postdoctoral work at the Van Andel Research Institute's Center for Cancer and Cell Biology. We've been fortunate to have her teaching at Hope since 2016. Her research focus is the biochemistry of the mitochondria, where many metabolic processes are centered and where cellular respiration takes place. I will be keeping an eye on the chat window. So if you have a thought, but don't want to interrupt, just send that chat if you think you might lose your train of thought, and I will catch it and be sure to jump in and ask at some good time. Or if you're just feeling a little bashful and don't want to jump in yourself. But I encourage you to do that, as does our presenter. And I am looking forward to hearing more about this. So without further ado, here is Dr. Kristen Dittenhafer-Reed, and learn about oxygen.
Thanks, Bruce. It's nice to see you all here virtually. I do enjoy really speaking to this group, but usually it's up in that HOSP classroom in Anderson Workman, but I'm glad that HOSP still was able to have classes in the virtual setting, and it's nice to see everybody here. As mentioned, I'm happy to kind of field questions as we go through, or we can save the questions and discussion period until the end of the time today. And so as Bruce mentioned, as you know from the course description, today we are going to be talking about oxygen regulation in cells. And this led to the Nobel Prize in Physiology and Medicine awarded last year, so in fall of 2019. Also, before we get any further, I do just want to say, if at any point there's any audio issue or something like that, maybe chat that to Bruce and let Bruce communicate that to me. I'll try to speak up and make sure that you can hear me okay. But if there's an issue, I want to make sure to know that. So what are we going to think about today? So today we're going to start off with a little bit of background about metabolism and the importance of oxygen. And then I'm going to walk through the three main discoveries that led to the Nobel Prize in Physiology and Medicine in 2019. So kind of stepwise talk about each one of these things and how they kind of all come together in the end to have resulted in that Nobel Prize being awarded. And then spend just a few minutes talking about specific applications of this Nobel Prize science to health and medicine. And then have some time at the end for both questions and for discussion. Bruce kind of nicely summarized this. We must have been on the exact same page. I was going to tell you a little bit about myself, but he kind of ran through all of these things already for me, so I won't waste more time on this. But I did just want to say that in the lab, we're really focused on understanding the mitochondria. I'm going to talk about the mitochondria today, the powerhouses of our cells, the metabolic centers. And so in the lab, we use basic biochemistry to try to understand how mitochondria function. And so I think about these metabolic things that I'm going to talk about to you all today a little bit quite often in my research, as well as in my teaching in biochemistry. So it kind of has aligned nicely. We're teaching metabolism right now in biochem. And so it's kind of been a nice overlap preparing for this talk as well. I do just want to give a shout out quickly to the students in my research group. The best part of my job is getting to engage with students at Hope College. And so for the past 4 years, I've had a lab of undergrads. Of course, last summer, as you can well imagine, our summer research program looked a lot different than what it normally does. And so here's just a picture of my small lab crew that we had for a few weeks when we were allowed on campus over the summer. Of course, masked and socially distanced. But normally this is what my lab looks like, and these are some of the great students that help me kind of do my research in the lab and things like that. So I'm looking forward to the day where we can take another picture that looks just like that. I did just want to ask a little bit about you, if you'd be willing. I know some of you have your cameras on and some of you don't. Maybe you know how to use kind of the reaction button on Zoom, and you're welcome to do that if you don't want to turn your camera on. But I'm just curious, as I think about how to present this talk, how many of you are involved or have backgrounds in science? I know this is the science and technology group, so I'm just curious, like how many people worked in science or might have been, have their background in the scientific field. Okay, so I see a few votes for that. And then my other question would be, similarly, what about medicine? Do we have medical professionals who are part of this crew? Okay, so a couple of medical professionals as well, or connections to medicine. That's great. And then finally, how many of you are just here to kind of learn something new and exciting? Okay, some new and exciting too. That's awesome. You guys are like Zoom pros. You're better than my students. That's great. Okay, so that's cool. Thanks for telling me a little bit about you. And so with that, we'll kind of just go ahead and dive right in. And so first, I just wanted to introduce you to the 3 Nobel Prize winners in 2019 for physiology and medicine. And so we see Dr. Kalin, Dr. Ratcliffe, and Dr. Semenza. These scientists each earned 1/3 of the Nobel Prize last year, but without each of their individual discoveries, the whole puzzle wouldn't have been put together. So each of them played a very integral role into the discoveries that led to the Nobel Prize. And you can see where each of them kind of have done their work that led to this discovery. Of course, as you guys probably well appreciate, these are lifetime achievements for these scientists, right? Things that they have studied for years and years and years that finally culminated in the awarding of this Nobel Prize. So specifically, they won the Nobel Prize for their discoveries of how cells sense and adapt to oxygen availability. And so let's unpack that a little bit. Their discoveries also are very important for both physiology and as well as pathophysiology or disease. And so as far as physiology goes, we can think about the importance of oxygen in metabolism, response to exercise, or the response to being at altitude, among many other things. And we also see a very important role for cells sensing oxygen in diseases like anemia or cancer or heart attack, myocardial infarction. And those are kind of the 3 kind of health applications I'll talk about briefly today. So the first thing that we have to understand in order to understand this Nobel Prize is why oxygen is essential. And so oxygen is essential to our cells because of its role in metabolism. And so I often show this crazy complicated map when I first introduce metabolism when I'm teaching, because one, it is a little overwhelming, but I think it's absolutely beautiful that our cells are literally catalyzing billions of chemical reactions at a time in our body. And somehow all of it's coordinated and all of it makes sense and all of it can respond so quickly to, let's say, changes in nutrient availability, or in this case, changes in oxygen availability. And so oxygen is really essential because of its need in cellular metabolism. So of course, we can look at cellular metabolism in these very complicated maps, but I like to often show metabolism in a much more simple sense. Okay, so we can think of it more simply than all of these billions of reactions that have to be happening in our bodies at one time. So what we're looking at here, we're just looking at kind of a schematic of a cell. And in cellular metabolism, we can say that food comes in, and the food that we're going to focus on for the purposes of today is we're specifically going to be thinking about glucose, sugar. So glucose comes into our cells, and through a variety of pathways known as catabolic pathways, or pathways that break a bigger molecule into a smaller molecule, we get to these cellular building blocks. And so examples of cellular building blocks include things like nucleotides or lipids or fats or amino acids. So we can break these things down, break glucose down into these smaller building blocks. And in the process, we get out some energy in the form of this molecule called ATP, or adenosine triphosphate. We'll return to that slide in a second, but for just a moment, let's talk about what ATP is. So this is really kind of can be thought of like the cash or the energy currency in our cells that helps to drive reactions that energetically usually wouldn't be favorable into a favorable state. So the hydrolysis of ATP is a very important energy source to drive chemical reactions inside of the cell. The structure, the chemical structure of ATP is shown here. This portion of the molecule is a nucleotide called adenosine, and then it has 3 phosphates, so 1, 2, 3. So that's where we get the name adenosine triphosphate from. This very last bond here, this oxygen to phosphate bond, is considered what we call a high energy bond. And what that means is that when that bond is broken off, we get some free energy that is allowed to be used to fuel reactions that normally wouldn't happen. So we have to have ATP around in order to do a myriad of different chemical processes in our body. Perhaps more simply, we can think about ATP as shown right here as the charged battery in our cells that's helping to fuel these reactions. Upon its hydrolysis or breaking apart to this molecule called adenosine diphosphate and then the free phosphate, we get some energy that we can use. And then we have a dead battery, and the dead battery has to be resynthesized to ATP. This recycling process occurs in your body many, many times a day, and I'll give you some specific stats on that in a few slides. And so this process is constantly occurring in order for our bodies to function. And all of this happens in the mighty mitochondria. Because it's my favorite part of the cell, I always have to give a shout out to it. And so this all is occurring in the mitochondria where we regenerate our charged battery to fuel reactions. Okay, so let's go back to our metabolic cell here. So we have glucose, it gets broken down into these cellular building blocks, and through that process, we get all of this ATP that we need to fuel different reactions. Now, very importantly, the ability to generate ATP requires the presence of oxygen. So if we were to get rid of oxygen, we could not generate ATP. Oxygen is absolutely essential in order to get this energy out. We also, in the process of metabolism, lose a little bit of energy in the form of heat. So if we think of this process as like an engine in your car, we put gasoline in our car. That gasoline is combusted in order to produce forward kinetic energy, right? We also lose energy in our cars as heat. We do that in our bodies as well, but we don't want to do it too much, just like you don't want your car engine to overheat as well. But some of this process is lost in the form of heat as well. We break all of these things down and generate ATP so that we can then build up other molecules inside of our cells. And so these processes that take cellular building blocks and make bigger things are known as anabolic pathways, and they require ATP. So we can take amino acids, for example, and build proteins or enzymes. This requires energy to do that. Or we can build cellular membranes, or we can replicate DNA and RNA. All of these things are needed if a cell is going to grow or divide. And so in a growing or dividing state, we need to burn our energy to make these buildings.
Quick question, if I may.
Yes.
If oxygen is required for the production of ATP, does that mean that for creatures that do not operate in an oxygen plentiful environment, they use something other than ATP as their cellular currency, or do they have a different mechanism of making it?
That's a really good question. So Bruce's question is, what about things that would grow in what we would call anaerobic environments, or places where there's not oxygen? On the next slide, I'll show you there are a few pathways in our cells. Maybe I'll just go there. There are a few pathways in our cells that generate ATP in the absence of oxygen, but they don't generate a lot of ATP. And so for us, we need a lot of ATP, so we use these other pathways. But certainly there are tons of microbes that can function in an anaerobic environment, or they do what's called fermentation. So we can't do fermentation. This is a process that leads to like carbon dioxide in beer and alcohol in beer, right? We don't do that. We can't make alcohol from glucose, but other bugs can. They have really different pathways. That's really good. So let's dig in just a little bit more specifically into metabolism. So here we're looking in yellow. The yellow outline is just like a blob of a cell. And now here's a picture of a mitochondria in red. Through the process of something called glycolysis, so you can think of that, the lysis of glucose, breakdown of glucose, we generate a molecule of that's called pyruvate, and we get just a few ATP out of this process, get a little bit of energy. That molecule enters into a pathway called the citric acid cycle, which generates really just one ATP, not a lot, a little bit. And then we produce these electron carriers. And so electron carriers are super important in the electron transport chain, which occurs here. And electron transport and ATP oxidative phosphorylation is where we generate a lot of ATP. So to give you a sense, per molecule of glucose, we get 2 ATP out of glycolysis, we get 2 ATP out of the citric acid cycle, and we would get about 28 to 30 ATP from oxidative phosphorylation. That's right. So these are big differences. And this pathway, as you can read through oxidative, right, we need oxygen in this pathway. And so this is where the oxygen comes into play. And so let's just look at that a little more in depth as well. So now we're zooming in specifically at the mitochondria. So from the last slide, now we're just looking at the mitochondria. And what I want you to focus on first is this kind of part of the slide that I am circling with my laser pointer here. This is called the electron transport chain. And so in this case, our electron carriers, which are known as NADH and FADH2, nicotinamide adenine dinucleotide, if you want the full words here, they served as electron donors into this electron transport chain embedded in our mitochondrial membrane. As electrons are donated through these different complexes, 1, 2, 3, 4, protons get pumped across the membrane. And so these protons get pumped across the membrane by these proteins that are embedded, these electron transporting complexes. And this proton gradient is super, super important. And the reason why this proton gradient is important is because it allows for the synthesis of ATP, by ATP synthase, which is shown over here. I'll explain that a little bit more in a second. And so what we find in the electron transport chain is, again, we transfer electrons from complex to complex. Through that process, we pump protons into the intermembrane space. This proton gradient is an electrochemical gradient. When that gradient is dissipated, we can use the energy to synthesize a molecule of ATP. And so this is how we get ATP in this process. One important thing, the electron transport chain requires oxygen, as shown here in this orange box. So oxygen is the terminal electron acceptor in the electron transport chain. If we were to remove oxygen, so if we were to get rid of this, we would essentially back up the chain, okay? So we would back it up, and we would have inefficient electron transfer, and we could no longer synthesize ATP. And so that's what would happen if we didn't have enough oxygen around, and that's why oxygen is essential. So this idea of an electrochemical gradient, if you've never heard of this kind of concept, or it's been a long time since you've heard about it before, I like to think about it as kind of a dam. So has anybody been to Hoover Dam? Anybody seen this? It's pretty amazing. I was there as pretty young, and I don't think I appreciated it, but it's a pretty cool thing. So if we think about this proton gradient as like Hoover Dam, right? All this water that's up here kind of being held back by the dam. These protons are being held back by the membrane between the inner and the outer membrane. And when we release that water, or we release those protons, like we release water through a dam, we know that we get energy from that process, right? So it's kind of a similar idea to try to understand ATP synthesis in the mitochondria that's promoted by this electrochemical gradient. Okay, so that's kind of the background section. So let's just give a quick recap of that section. So oxygen is required for cellular metabolism, and specifically it's required for the synthesis of ATP. And ATP is this essential molecule that fuels reactions inside of our cells. Just to give you a sense, give you a little stat about ATP that I think is really interesting. The average human uses their body weight in ATP every day. Okay, so a 150-pound person would use 150 pounds of ATP every day. This doesn't mean, right, that you gain 150 pounds, you lose 150 pounds, right? We know that that's not true. It's all this recycling process. And so each ATP molecule through this charged battery, dead battery cycle is recycled about 500 to 750 times a day. Now that's an estimate, but that's what people think happens. And so again, if we think about the necessity of oxygen for this process, and that it needs to happen 500 to 750 times a day, we now see why oxygen is so important for us. But oxygen levels are not always constant. And so there are a number of things that we can relate to when oxygen levels change. So exercise, right? When we're exercising, for example, our muscle cells might not get as much oxygen as they need to get. When we're at altitude, we're just at a lower percentage of oxygen in the atmosphere than when we are in Holland, Michigan. Certain illnesses lead to reduced levels of oxygen, heart disease, stroke, anemia, or cancer. And then finally, cell location can also have a big impact. So a cell in your body that's closer to an artery or a vein carrying oxygen-rich blood will have more oxygen than something further away. So because we have this situation where our oxygen levels are not always constant, our cells must be able to adapt to changing oxygen level, but still provide this consistent output of ATP. So hopefully I've made it clear, like we can't just lose a bunch of ATP when we don't have oxygen. Somehow our cells have to say, okay, we have to figure out a different way to do this. And how do cells do that? And so that really is the crux of the discoveries of the 3 scientists I presented at the beginning who won the Nobel Prize in 2019. So with that, now I'm going to transition from the background of oxygen being essential into the 3 main discoveries that led to the award of the Nobel Prize. There are 2 words that I'm going to use as I talk through this. And so I just want to introduce these definitions so that it's clear. So the 2 words that I'm going to use, the first is Normoxia, and that means a normal level of oxygen. So that would be, if I was growing cells in my incubator here at Hope, I would grow the cells in the presence of about 10 to 20% of oxygen, so near atmospheric levels of oxygen. The other word that I'm going to use is hypoxia. So hypoxia equates to low levels of oxygen. Again, if I were growing cells, I would be growing those cells at about 1 to 5% oxygen, so much lower. Now, I tried to look up more specifically like what the intracellular concentration of oxygen would be like in a human. And I just don't know that people know those numbers. So I don't know exactly what that equates to as the intracellular concentration of oxygen. I think that's a much more difficult measurement. It might be out there, but I didn't come across it. So just to give you a sense of oxygen levels that we're talking about. The first thing we're going to talk about is what happens during hypoxia. And this is really what Dr. Semenza, one of the Nobel Prize winners, was really focused on. Kind of his career was dedicated to like what happens in the hypoxic response of a cell. The first thing that happens when your cells are experiencing a low level of oxygen is a rise in this protein hormone called erythropoietin, or EPO. Now, some of you might be familiar with EPO. If you follow cycling at all, it's been some time since Lance Armstrong won a bunch of Tour de France's, but he is famously known for kind of doping scandals, and a lot of that doping had to do with the use of EPO to kind of artificially enhance exercise capacity. But EPO is a really important thing inside our body, and what EPO does is it's a hormone, and it signals to increase levels of red blood cells. The more red blood cells we have, the more oxygen we can carry around our body. So one way to get at dealing with a low level of oxygen is to increase the amount of carriers we have for oxygen in our body. More red blood cells, more oxygen transport. But the question was, how is this process controlled by oxygen levels? So what is the true sensing mechanism that says, okay, I have low O2, but now I need higher amounts of EPO? What's the sensing mechanism there? And that's really where this first discovery comes in. So Dr. Semenza discovered this protein called hypoxia-inducible factor, which I will call HIF for the rest of the talk. He discovered HIF, and he found that there are regions of DNA next to the gene that encodes for EPO that's directly regulated by oxygen levels. So in cells with low oxygen, they saw more EPO that had to do with the transcription of this gene of EPO. So I'm using some words, again, some of you, depending on background, might be super familiar with. Some of you might be like, man, I haven't seen that in a long time. So let's just break this down for a second. So this is the central dogma of biology. And I'm going to be using words like transcription and translation quite a bit. And so let's just clarify what that means. So we have DNA inside of our nucleus. It holds all of our genetic information, our genes and the regions around those genes that say whether that gene should be transcribed at high levels or low levels, things like that. We can think about DNA as being the blueprint for our whole house. And so if DNA is, again, so we have a blueprint for a whole house here. That's like our DNA. Through the process of transcription, we get these regions of our genome that are transcribed into what are known as messenger RNAs or mRNAs. So that would be like me saying, okay, I see I have this whole blueprint of the house, but I really just want to make the door right now. So I just want to get that specific region of the genome that's talking about how to make the door. Through the process of translation, this is where machines come in, read the code that's embedded in the mRNA, and actually synthesize the proteins. So back to the door example, this would be like taking the blueprint for the door and building the physical door. The physical door is what does something. The blueprint didn't do anything, but now we have the physical machine of the door. So I also keep talking about proteins a lot. And I know that our first thought when we think about proteins is we think about this, but we're actually thinking about machines inside of our cells, things that when we do pretty pictures of look something like this. They have a unique structure, and that structure drives its function. Proteins are made up of amino acids. So chains of amino acids polymerize here to form this, what's shown as a blob of a protein here. For those of you more chemically oriented, our amino acids have a very consistent structure with an amino terminus, a carboxyl terminus, and then this unique chemical group called an R group that dictates what the chemical properties of that specific amino acid are. So we have a number of amino acids in our bodies, and the way they connect and the way that protein folds drives its function. So some proteins function as signaling molecules. Some proteins are enzymes that act as catalysts for all of these billions of reactions that have to happen inside of our cells. So with that, let's get back into HIF. Okay, so DNA segments next to the EPO gene are regulated by oxygen levels. And what was found by Dr. Semenza is that this HIF protein binds to the DNA segment in an oxygen-dependent manner. So when HIF binds to gene regions next to the EPO gene, it signals the EPO gene to be transcribed, or for that little piece of the blueprint to be made into the functional unit of EPO. What Dr. Semenza found was that low oxygen meant that there was more HIF around. And another really important thing about this discovery is that it was a ubiquitous mechanism. So what I mean by that is that it wasn't just in one cell type or one disease or one context. It seemed that the majority of cells were responding in a very similar way to a lack of oxygen. Now that's always a really cool thing in biology or biochemistry because context-specific responses are important, but when we can find these master or overall responses, that's really exciting and really impactful. Let's talk about HIF just a little bit more. So HIF is what I would call a master transcriptional regulator. So HIF is known to activate the transcription and production of at least 40 genes and proteins. We can look at this picture to see what HIF is doing. So here we're looking at a cell in hypoxic conditions. So oxygens are crossed out. This HIF protein under hypoxic conditions comes in the nucleus, binds specific regions of DNA called hypoxia responsive elements, and then leads to the transcription of a number of different target genes. Some of these genes, as we already discussed, are like EPO, and they're involved in the synthesis of red blood cells. Some of these genes are involved in metabolism, so cells actually Hello? I just got kicked out of Zoom and back. Weird. Okay. Hang on. Okay, can you see my screen okay? Yeah? Okay, sorry about that. That was odd.
Yep.
Okay, so I was saying as it went out, cells actually can rewire their metabolic processes to kind of build up, like, for example, the glycolytic pathway, which doesn't require oxygen, but still allows us to get some ATP through that metabolic process. And another thing of interest is that HIF also has a role in what's known as angiogenesis, or the synthesis of new blood vessels. And so if we want to get oxygen more places, more blood vessels to carry those red blood cells is important. Overexpression of this HIF protein. So yes, Bruce.
So there is something about these HIF proteins in common with other such things that serve to signal the transcription of this DNA. Is that accurate?
That's correct.
Yep. DNA gets transcribed for a bunch of reasons. This is one of them.
Exactly right.
Do these things that induce transcription of DNA have a common shape or structure? Or is each one unique and this just happens to bind here, and the thing that makes it special is where it binds as opposed to what it looks like on the outside?
Right. So I'm going to try to answer that. You let me know if it's not what you're going for. So we see down here in this picture this thing called an HRE, a hypoxic responsive element. Those are specific regions in DNA, and they have specific sequences that HIF likes to bind. So the actual sequence of that DNA, that transcriptional element or the promoter element, kind of says, okay, HIF, you would bind here. So if we looked at all 40 plus genes that HIF controls, we would see a similar sequence upstream of it where HIF wants to bind and then says, okay, transcribe X, Y, and Z gene. Does that make sense? Is that what you're going for?
Yeah, so there is a common mechanism for the transcription of DNA. The thing that makes HIF operate specifically as it does is the site on the DNA that it binds to, right? Mm-hmm. But the machine that actually does the transcription, right, doesn't know that it's transcribing HRE. It just sees, hey, there's a HIF here, right?
Exactly right. So we can think of HIF as like a recruiter for the machines that have to do transcription. So they bind somewhere on DNA, and imagine that's like waving its flag and say, hey, come here, transcription machinery, and transcribe this gene. And so then we get more mRNAs, and then that leads to the production of proteins that are involved in all of these.
Okay, so to restate my question, do the things that bind to DNA all wave the same flag and they just stand on different places? Or are there a different flag for each of these proteins?
It may be an oversimplification to say this, but I'm going to say they all wave the same flag.
Gotcha.
So HIF is a transcription factor. We have loads of different types of transcription factors that all respond to different cellular signals. So, you know, you might have a signal that responds to increased glucose levels in your cells. That goes and binds to a specific region of DNA and waves its flag and says, hey, come transcribe these genes. The transcriptional machinery itself is all the same. So the machine that does the transcription is all the same machine, no matter what transcription factor you have, saying, hey, you should transcribe here, or hey, you should transcribe here.
Gotcha. Thanks.
Cool. Yeah, good question. So with all things, we have a balance. So we do want HIF in times of oxygen depletion, but we also see overexpression of HIF in a number of disease contexts, including cancer and tumor invasion, things like cell survival and proliferation, which can be good, right, in an embryonic development or in a normal state, but can be bad when we're thinking about something like cancer. We also see HIF being implicated in a variety of inflammation and inflammatory diseases. Okay, so the first discovery was of this transcription factor called HIF. And the discovery was that HIF binds to specific regions of DNA and controls the transcription of a large number of genes that were important when oxygen levels decrease. But what wasn't known was how are HIF levels controlled. So we still don't have a precise mechanism of how oxygen is controlling HIF levels. So importantly, the second discovery here was trying to understand how HIF levels are controlled. And what was found was that at normal oxygen levels, or at Normoxia, HIF becomes rapidly degraded by something called the proteasome. And we can just think of the proteasome as the recycling center of your cells. So sometimes we have old proteins and they need to be taken care of, or old lipids, they need to be degraded, things like that. So we have ways to get rid of these things inside of our cell, and the proteasome is one of them. So here's a picture. There's a lot going on here, but let's just, I'm going to show a few things. So the blue thing again is HIF. That's that protein that binds to DNA. In normal oxygen levels, we don't need HIF. We don't want HIF to be telling us to make more red blood cells and doing all these things. We don't need it to be there. So we need HIF to go away. And the way HIF goes away is that it's broken down through this thing called the proteasome. And so the proteasome chops that protein up into individual peptides or proteins. And specifically, the way this HIF protein is targeted to the proteasome, so proteins don't just go to the proteasome to be degraded. There has to be some signal to say, hey, this thing doesn't need to be here anymore. And the signal, which is shown in this picture in blue, is the addition of these chemical changes, chemical groups called ubiquitin. So we can just think of these as chemical modifications that occur directly to that protein. And much like the transcription factor can be a flag for the transcriptional machinery, this ubiquitin group modification is a flag to say, take this protein away. We don't want this protein anymore. Break it down, recycle it. We don't need it. And so we could talk in depth about what ubiquitin is later if you have questions about that. But for now, let's think about just as a chemical tag. That tag gets put onto the protein, and then that protein is degraded and taken out of our system. But again, this was nice. This is a good, awesome discovery, but the connection to oxygen still isn't there. So they knew that this was happening at Normoxia, but they didn't know why. So what specifically led to the degradation or the addition of ubiquitin? It all comes into play, this little bean protein over here. This little green protein is known as the VHL protein, or the von Hippel-Lindau protein. It's a mouthful. And so this VHL protein, as shown in this slide, when VHL binds to HIF, it promotes the ubiquitination of HIF. Okay, so I'll say it one more time. So we got a lot of molecular things going on here. We introduce a new protein to the party, and that protein is VHL, or von Hippel-Lindau. I'll talk about it more specifically in a second. And VHL, when that green bean binds to this HIF protein, as shown here, it signals its ubiquitination and its degradation. So what is this von Hippel-Lindau protein? Just to put it in context, this was Dr. Kalin's discovery, the second person awarded the Nobel Prize. He was studying this disease called von Hippel-Lindau disease. This is a genetic disorder that afflicts about 1 in 30,000 people, and it's due to mutations in the gene for the VHL protein. So mutations in this gene then cause defects in the protein's function. Again, it's genetic, and it was shown to lead to an increased risk of a number of different cancers. And it also leads to the development of tumors or growths that look largely like kind of newly formed blood vessels. The discovery here was also that the presence of VHL prevents the onset of cancer. And so when VHL was normal, the cancer incidence was lower. And this scientist did a bunch of studies in cells and found that cancer cells that lack the VHL gene express very high levels of our hypoxia-regulated genes. So when VHL went away, HIF kind of was out of control. HIF levels are no longer brought down because the protein was degraded. And so these cancer cells expressed high levels of the hypoxia-regulated genes. And as all good science, we want to always try to like rescue this phenotype. And so Dr. Kalin was able to reintroduce healthy VHL into these cancer cells and saw that the gene regulatory mechanisms kind of were brought back to normal. So no longer was there this induction of hypoxia regulated genes. So let's just talk about this one more time. So what does this mean? All of these things mean that the VHL protein is required to label proteins, one of them being HIF, that signal HIF for degradation. So the VHL protein here was really essential in order to say, okay, we don't need you, HIF, anymore. All right, so the very last piece of the puzzle. We still haven't connected oxygen directly into this mechanism. We've made observations about when oxygen levels change, what happens, but no direct regulation, no direct mechanism has been shown here. So the last question was, how does oxygen regulate the interaction of this protein VHL and HIF? So I'm going to go back to my door example for just a minute because I need to introduce something new. So I told you that through the process of translation, we synthesize our proteins, which are like our functional door. We have a whole nother layer of complexity inside of our cells, which are known as post-translational modifications. So once the protein is made, chemical changes can be added onto those proteins here, and those chemical changes then drive functional changes. And so just to connect my research a little bit, this is what I study in my lab. And so I study how the post-translational modification of proteins and enzymes alters their function. And so we can think about it in the door context of like, okay, well, we have this door, but we could add a mail slot, right? And that changes the function of the door. We could take away the doorknob. We could add windows. We could change its color, right? We can do subtle things to our proteins that then change their function. And so what the last discovery was of a post-translational modification that controls the interaction of VHL and HIF. And this post-translational modification is called Prolyl hydroxylation. Again, for those of you who are interested in structure, here is a proline. A proline is a specific amino acid. So we see these prolines found inside of the protein HIF and other proteins too. And through prolyl hydroxylation, we can see that a chemical change has been made. So here we do not have oxygen, and here we've added what's called a hydroxyl group, an oxygen and a hydrogen. This process is catalyzed by enzymes called prolyl hydroxylases, or PhDs. Wouldn't it be fun to get a PhD on a PhD? I think that would be cool. Anyway, see, these are the corny jokes I tell in class. They sometimes hit, they sometimes miss. But anyway, so we get these hydroxyl groups that are put onto prolines. We're going to go back to the bigger schematic now. Now we see kind of a direct involvement of oxygen, right? We at least see an oxygen molecule being present in this process. So what was found was that oxygen levels lead to the hydroxylation of HIF. When HIF is hydroxylated, this allows VHL to bind, and then it drives HIF degradation. So let's look at it pictorially. We're going to walk through each one of these steps here. So in the presence of oxygen in a normoxic setting, we have oxygen around, and these prolyl hydroxylases, or PhDs, put hydroxyl groups onto HIF at specific proline amino acids. That hydroxylation of HIF then signals the protein VHL to bind. And when VHL binds, HIF then is degraded. So this is the mechanism of how specifically under normal oxygen levels, the HIF protein is signaled to go away, and how HIF is therefore responsive to either normal oxygen or low oxygen levels within the cell. And this really, I'm going to put it all together in a slightly different picture here. This is really what the Nobel Prize was awarded for in 2019. It's kind of this collection of discoveries that led to an overall understanding of how cells sense and respond to either the Normoxia, where HIF needs to be degraded, or to hypoxia, where HIF needs to come into the nucleus and to do its job to elicit all of these responses to change the transcription of genes we need when we don't have oxygen around. So again, this is none of my work. These are all these great Nobel Prize winners' works, and each of them here are assigned to kind of their discoveries and their impact on this process. And so kind of to wrap this section up, this is what the Nobel Prize was awarded for. This big sensing mechanisms that most of our cells have to respond to the high or low oxygen levels. With that, I want to transition a little bit to like, why does this matter, or why is this important in human pathology and pathophysiology? I showed this slide at the beginning, but of all the impact that these oxygen sensing has when we think about human health and disease. First, we're going to focus on this proline hydroxylation step and how altering this step is used to combat different types of disorders. So again, there are these enzymes called prolyl hydroxylases, and people are really interested in developing drugs that would inhibit prolyl hydroxylation. If we can inhibit prolyl hydroxylation, we actually would increase HIF-1 alpha levels. Okay, so we kind of fake our cells into believing they're in a hypoxic state when really they have plenty of oxygen around by inhibiting the process of prolyl hydroxylation. So where would this be beneficial? So this would be beneficial in the context of like anemia. So in anemia, we have low red blood cell levels, low oxygen levels. If we can trick our cells into thinking that they need to be making more of the genes and proteins required at low oxygen levels, this would be a really good thing. And so indeed, people have done this by developing inhibitors that specifically inhibit prolyl hydroxylation and then rise elevate the amount of HIF-1 alpha, and then that subsequently leads to an increase in EPO. There are a few molecules shown here that are used in the clinic. I thought this was interesting. This was the most recent one that I could find. This little blurb was published in February of 2020. And so this is a new drug application for this drug here to use for the treatment of anemia that's associated with different types of disorders. So indeed, this pathway, the elucidation of this pathway has allowed other scientists and doctors to create, not create, but to learn ways that we can interrupt this pathway and be helpful in human health. So that's one example where we want to have HIF around. We want to stabilize HIF. It's a helpful thing. But there's a counterexample in cancer. So in cancer, we often see very high levels of HIF. So it's kind of small here, but I'll highlight it. A number of cancers are characterized by an increased level of the HIF protein. Now, why is that? So we know that cancer is a tumor, right, is growing and dividing rapidly. In this process, we have a number of cells in a tumor that are kind of exposed to the vasculature and have enough oxygen. But on the interior of a tumor, as shown here in blue, we have a number of cells that are oxygen limited. And so in order for those cells on the inside of a tumor that are away from vasculature to be able to kind of keep doing their thing, they need some sort of way to deal with this lack of oxygen. And one of the ways some types of cancers deal with that is by increasing the level of this HIF protein and essentially faking the cell into thinking that HIF should be there and that all of these genes should be developed. So what's the outcome of this in the context of cancer? Well, increased genetic instability arises, right? We have more cells dividing when they shouldn't be, genetic mutations, things like that. Some of you may be familiar, there's a very big and amazing push to use kind of your own immune system to combat cancers and tumors. And it seems that this type of HIF expression is related to a poorer immune response. And this also leads to less susceptibility to chemotherapy. Now, part of that is because, right, we just don't have vasculature to deliver things to the inside of the tumor, but it also has to do with kind of this HIF overexpression. So here's a case where we actually want to inhibit HIF, and we'd want to inhibit HIF specifically in the context of the cancer cell. This next slide presents some drugs that are used in the clinic or are being researched as ways to prevent HIF accumulation. So here's our protein HIF here. And so there are a number of different ways that people have tried to do this. Some of these are FDA approved things, but others aren't. As you notice, there's nothing here that's like directly inhibiting HIF, right? The perfect inhibitor would target HIF directly. People haven't found ways to do that. These are all what I would call upstream inhibitors. So we're like affecting something that eventually affects HIF. That's not the best way to fight a disease, right? You want to hit the direct target when you can. That can be really difficult. So again, here in cancer is a context where we want HIF to actually go away. The last example that I'll tell is about ischemic heart disease. So in ischemic heart disease, right, your arteries are slowly narrowing. And so this leads to less red blood cell transport, which therefore is less oxygen transport. So in this case, right, we have hypoxic conditions. And in this case, HIF actually is important. So we see HIF overexpression. It is shown to be connected with cardioprotection. So increasing our HIF levels allows for some cardioprotection, especially in the context of like a heart attack situation. And so in this case, right, we would want drugs that promote HIF stabilization. So this is more similar to the anemia context where HIF is good. And so certainly there are clinicians that are looking into ways to be able to do this in a heart attack. The very important thing that I should say, though, is that this is a balance. So in heart conditions specifically, we can't just say we want HIF there all of the time. This has actually been shown to be detrimental as well to a cell. So it's this delicate balance of how much HIF should be there versus not that scientists are really trying to understand in order to kind of harness this pathway for therapeutic purposes. Okay, so with that, that's all I have for today. I just want to say thank you for listening and for being here on Zoom. I appreciate it greatly. I really enjoy talking to this group. And I would be happy to take any questions, or if you guys want to have any kind of post discussion, I'll do my best to answer. You guys, I was just talking to a colleague and we were saying, man, they always have such good questions. We get nervous about it. So, but I'd be happy to answer anything that I can.
Is the biochemistry that you've described throughout this talk ubiquitous to all animals, cold-blooded, warm-blooded, whatever?
Yes, I'm going to say yes. Yep. So I would say all mammals. All mammals. I would be very confident in saying that. Yeah.
And this other thought that occurred to me was during hibernation, do we have any knowledge about APT production when animals are hibernating?
There is some knowledge in that, and people actually really study that. They think it's really interesting because those animals still have to make ATP. They still have to function. They're just in like a very reduced metabolic state. So I think that in hibernation, animals turn to fat stores. And so they burn fat instead of glucose, and that fat really provides an abundance of ATP. Hibernating animals, we have two types of fat in our bodies, white fat and brown fat. As adults, we don't have a lot of brown fat. As humans, really, we don't have a lot of brown fat unless we're babies. And that brown fat has more mitochondria in it than white fat does. And so this increase in brown fat and then the degradation of that, I think, kind of helps with this ATP production process. And also, there's an aspect of like keeping the animal warm in there too, but we won't go there.
Thank you.
Be sure to unmute yourself if you have a question.
Could I ask a question?
Absolutely.
I'm somewhat familiar with post-translational modification of proteins, specifically methylation and hydroxylation of lysine. I've never heard of hydroxylation of proline before. This is a proline hydroxylase, proline hydroxyl synthase, perhaps. That is it very specific for HIF protein, or is it a more general? Does it act on other proteins as well? How is its activity regulated?
That's a really good question. So yes, prolyl hydroxylation does happen more than just to HIF. So there are other proteins that get hydroxylated at proline, but it certainly, in my opinion, it is not as abundant as the other modifications you talk about, like methylation, or like in my lab, I study acetylation and phosphorylation. These things we see much more frequently than we see prolyl hydroxylation itself. Prolyl hydroxylases, and this is going to get in the weeds a little bit, they're actually regulated very interestingly. There's a very interesting connection between a metabolite that's made in the mitochondria, and it kind of feeds back to the nucleus and regulates the activity of a prolyl hydroxylase. And so there's more connection there, at least in the metabolic context, for how prolyl hydroxylases themselves are controlled in their reaction.
This gets into a little bit of stuff that you may not want to hear about, but about 55 years ago, I did a PhD on the response of bullfrogs to low oxygen, sort of trying to understand how they reacted to high altitude and like that. But this was way before all this biochemistry was understood. And so I was basically looking at the differential synthesis of different types of hemoglobins in these frogs. The problem, I went on to other things, and so I never really solved the problem. But this work brings a whole new way to look at interesting problems that have been around for a long time. Appreciate
Certainly.
it very much.
Yeah, thanks.
This may be an odd question, but why does post-translational modification even exist? You would think if you wanted a door with a mail slot, then you would have DNA to code for a door with a mail slot, rather than creating a whole nother mechanism in order to do that. Is it because the post-translational modification has to happen somewhere other than the nucleus, and it just needs to be local, like at your local Walmart, as opposed to the great big, shipping center where the doors are manufactured? Or can you shed some light on that?
Yeah. So in my opinion, there are a few different reasons why post-translational modifications exist. So we can only store so much information in our DNA, right? We have to fit all that DNA into our nucleus. And so essentially the ability to post-translationally modify and regulate a protein limits the number of genes that we need to have, right? So I don't need to have a gene for a door with a mail slot, and a gene for a door with a window, and a gene for a door with a purple doorknob. I need one gene, and then I need 1 or 2 proteins that can tell that protein when to change or when to do something different. So that's one reason, right? We reduce the complexity of what needs to be encoded within the DNA. The second thing is, just as you say, it could be that, okay, we need that protein only to be modified when it's in the mitochondria, which turns that activity on. And so it could be a spatial thing, right? We need that protein to be active here and inactive there. And then the final thing would be temporal or timing. So post-translational modifications, we can turn something on and off quickly. Whereas if we want to change the amount of something from the DNA level, we have to wait for transcription to occur, or for the signal for transcription to occur, then transcription, then translation, then the protein to be made. And then if we don't want that protein there anymore, we have to wait for that protein to be degraded. So I think it's a faster way to control inside of the cell what's happening. So those would be my kind of 3 reasons why those types of things exist.
But that introduces whole new levels of complications, controlling of the proteins that do the post-translational modifications and so forth. I've been particularly interested recently in the epigenetics questions regarding the post-translational modification of histones. Mm-hmm. And that just gets into a morass that we're years away from solving.
Yeah, definitely. I was trained, my lab in graduate school was all a post-translational modification histone lab. And so, yeah, it is, right? It does add a whole nother layer of complexity. And again, I try to impress this on my students all the time, and I don't know if it's lost or not, but to me, it's so fascinating. It's one of the reasons why I love science and why I love this stuff is like, how does it all work? And how, it's just totally amazing.
You mentioned on one of the slides you had I think a HIF alpha and a HIF beta. Mm-hmm. Is there anything we should know about HIF beta?
Yeah, so I took that out because there are a lot of alphabet soup already happening. So HIF1 alpha, there are different versions of HIF, and it actually has to kind of bind to another component of that complex in order to do its whole job. So there's kind of 2 pieces of HIF, and they have to come together in order to then get the response at the DNA. Any other thoughts? Any other questions? Want to know what's going on campus? Yeah, I don't know.
We do have a few minutes left if anybody does have anything else to discuss. Otherwise, I suppose we could wrap the class early. Bruce, do you have any thoughts on? You're muted.
I hate to ask yet another question, especially one off in the weeds, but I will give it a stab.
Okay.
I am amazed by how all this molecular interaction happens, given that there are only so many molecules that a given molecule can be in some sense adjacent to be affected by. Right? And the chance of having the right molecule be close enough, right, to do that phosphorylation or to do that whatever needs to be done just seems so unlikely, given the vast number of molecules that are sitting around in our cytoplasm waiting to do something, that it seems like it's almost got to be a quantum effect in order to have the range necessary to have enough neighbors to work. Does that make any sense at all, or am I completely off in the weeds?
No, I hear you. And I don't have a good answer to that question. I do think we can think about like the amount of things inside of our cells. Like, I don't know what the chance encounter of one, right? So we talk about the most efficient enzyme would be like at the diffusion limit, right? Just how quickly those things can come together and apart. And so our cells kind of combat that a little bit by having compartments, right? So like there's lots of things that happen inside of the mitochondria. So at least all of those molecules are within one place and those enzymes are in one place. We have the nucleus, right? We have all these different compartments. In order to do that, we have some enzymes that are like, what do we call them? Multi-component, I guess would be the right word. So those things catalyze not just one reaction, but let's say 3 reactions all combined in one. So you can imagine like it's a little substrate channel, right? So that molecule goes from one place to the next without ever having to leave the enzyme and come back. And not all of our enzymes work like that, but some of them do. But yeah, it is rather amazing. And you also then have to think about like, what are the relative concentrations of these specific molecules in these specific regions? If you have a high enough concentration, the likelihood that X and Y bump into each other is better than if you have a low concentration. So no, it's a really great question.
The infinitesimal items of everywhere are universes in themselves, just happening without understanding.
Well said.
Can I ask about the binding of oxygen to the hydroxylase, right? Is this a heme protein?
That's a really good question. Give me 1 second. I don't think so, but I can tell you that in a second.
I'm not aware of any other binding of oxygen specifically to enzymes.
Yeah, you're asking a very good question. I should know the answer to this. I don't think it has a heme. I haven't thought that deeply about that. That's a really good question. I'm sorry I don't know the answer to that off the top of my head.
No problem.
Yeah, I don't know. Yep, no, they don't have hemes, but I can't find how.
Is it structure is solved?
Yeah, it looks like that. It looks like it might have an iron sulfur cluster. You're familiar with? It looks like it might have an iron sulfur cluster. You're familiar with that? Yeah. All right. Well, I guess if there's no other questions.
Excellent presentation. Thank you very much.
Yeah, thank you. Again, I really enjoy this. if something comes to your mind that's burning, feel free to email me. You can find me on the chemistry website at Hope. And I'm happy to try to think about that a little bit more.
Thank you very much. It was wonderful.
Yeah,
It was great.
Thank you.
Appreciate it.
Thank
Yeah.
you.
Yep.
Okay.
Thank you. It's good to see you all.
Thank you.
All right. Well,
Thank
hang in there through an interesting week.
Indeed.
Absolutely. Thanks for taking the time to
Yeah.
come on. I think she's gone. And Bruce, thank you for leading this, for coordinating it. And I owe Bill Wells an answer. I took a note and then I walked away. Okay, thank you so much. You all have a nice afternoon.
I will let you know. I will let you
Oh,
know
there
what
you
I
are,
hear from Hector. I will let you know what I hear from Hector.
Okay, great. Thank you, Kristen.
Sounds good. Thanks, Bill.